Okay. Now, remember, in the last class, I promised you to show you few more codecs, but those two codecs are pretty important. Before going to understand those two codecs, for example, one is Fountain codec, another one is Yin Yang codec. So I thought to now cover certain basics in biology, right, so that you understand them much better way, right, and what are the really needs in terms of codec. So today, I'm going to give you some basic background about some historical events that happened in biology. And after that, I'm going to talk about certain bio operations that you can do with the DNA actually, okay? So I think, and then of course, while telling you the history of biology, uh, you will see that, uh, there are many interesting events that has happened. And I will also show you some I will take an specific example of HIV, and I will show you how that infection works. So it's really interesting thing to watch this. We call it secrets of life, and I think which is true, right? The secret of life is there in biology only, right? So basically, uh, as I will mention towards the end, there is a course Secrets of Life which you might have seen by Eric Lander, on EDX. Uh, what was known in India about biology? I have not spent time in collecting that history, so I'm starting from, normally I also look at in India, what happened in India. But anyway, so I would say that microscopic biology started in 1665. By Robert Hooke. Uh, you know, who first time using microscope, he observed that organisms are made of cells. Okay, in Hindi you call it koshika, right? You might have read about koshika, right? Cell. Now, so from 1800 to 1870, so basically once the microscope was discovered, people started looking at small things. And 1865, Gregor Mendel discovered. So I think from 1800 up till 1830, a lot more discoveries happened. With respect to cells. Once Robert Hooke observed it, many other people started observing things in biology. But 1865, Gregor Mendel, discovered the basic rules of heredity of garden pea. So you might have remembering this pea experiment. I don't know in your biology. I will tell you why this happened. This pea experiment happened because of a bacteria, right? So you see the connection. Interesting history. I mean, it's a very interesting thing. I will tell about more in between. Then 1869, John Frederick discovered DNA first time, deoxyribonucleic acid, and named it nuclein. This is something 1869, right? Then 1881. So in this period from 1880 to 1900, couple of major things. 1881, Edward showed that chromosomes are composed of nuclein. So nuclein, he was calling DNA as a nucleon. Okay, and chromosomes. Why it is the name is chromosome is because color-like objects, right? So chromosomes. Now we know that we have 23 chromosomes, right, in humans. You might have knowing this thing. 1899. Richard renamed nuclein to nucleic acid. They call it, then he called it as a nucleic acid. Okay. So this is very early history. Then by 1900s, so from that 1830s and so on, finally till 1900, chemical structures of all 20 amino acids had been identified. You might have heard that doctors advise you take proteins, right? Because proteins if you look at in detail, proteins are made of amino acids. So those are nothing but nutrition things because they are the building block of life. So if you have amino acids, you can make any protein. So the body can make because you need enough raw material to make some sort of protein, right? So it's very, very important. So that's why doctor advise you take amino acid. You take protein, right? It's very there are protein drinks as well, right? Available nowadays. Anyway, so from 1900 to 1911. So in 1902, Emil Hermann showed amino acids are linked and from proteins. So basically, this connection was discovered. That amino acids. So basically, the proteins are nothing but so building block of protein is nothing but amino acids. So you can actually make them out of. See, basically, so protein, if you observe, it's like a 3D structure. So if you look at a hemoglobin protein, right? So which what is the purpose of hemoglobin? To carry oxygen, right? So different proteins do different kind of job inside your inside your body. Okay, so these proteins are made of amino acids. So this connection was discovered basically in 1902. Then in 1911, Thomas Morgan discovered genes on chromosomes are the discrete units of heredity. So basically, gene, what do you mean by gene? Gene is a instruction for making protein. Okay, if you look at the DNA or the genome of any individual, so genes are nothing but instructions for making protein. So stretches of DNA inside your genome. Okay, so normally in the human genome, which is around 3 billion base pair, only 2% accounts for protein building instructions, and rest of things are different things, architecture. It's a very beautiful in terms of from a computer science point of view. Okay, then another nucleic acid was discovered in 1911. So Aaron discovers RNA, ribonucleic acid. It's a very beautiful thing, and I think I love RNA more than DNA because people say that how life began on this planet. So actually, RNA was formed because of some other chemicals. First formation of some other chemicals lead to RNA. Now RNA is very volatile molecule, so it cannot. So basically, it's very unstable kind of thing. So RNA actually requires some sort of storage mechanism. That's why DNA was discovered. So DNA is actually so nature has created DNA for storing information. So RNA evolved into DNA basically. Okay, that is how things are. And why RNA important? Most of the deadly viruses that you see, COVID-19 or HIV, they're all made of RNA. So RNA is a very, very important thing. If you understand about RNA, you can discover many things. Okay. So RNA is actually very, very important, right? So, and I think initially people thought that, okay, so we have RNA and then we have DNA. So I'll tell you about what is commonly believed, that central dogma of biology is something very common. So from DNA, so what happens? Information flows from DNA to RNA and RNA to protein. Okay, this is a common belief. Something around 60s it was observed, but now there are things turns out to be much more complicated than such simplicity. But anyway, I will explain that phenomena in detail. Okay, so in 1941, George and Edward identify that genes makes proteins. So again, so very precise thing that there is some hereditary material on the genome which gives you protein. Okay, so this process of central dogma is observed basically. Okay, and also we use quite often this, that A is complement of T and C is complement of G. That was observed in 1950 by Edwin. He finds that cytosine complements guanine, C is complement to G, and adenine is complements to thymine. So A is complement to T. This observation happened in 1950. see such a early stage of biology, basically, 1950. And by the time you know that, I think atom bomb was discovered already, right? So physics has progressed a lot. Even computer science progressed by 1950, right? In 1960s, early computers, and I think you know that personal computers started in 1970s or so, 77, right? So, very, very early stage. So we are just about, we just started learning few things about biology, okay? So our information about biology, even though we have, say, 10,000 years of, I mean, at least the history that you can go back, but we have very little information about biology, actually, okay? Then the era of 1950 to 1952. So Bush discovered in 1950, you know, how to isolate tRNA. So why this word tRNA? Stands for transfer RNA. It's a molecule which involves in central dogma process. I will tell you what is central dogma and how it is useful. tRNA is kind of a structure. Again, see, tRNA is, again, you can look at a secondary structure form. So, and then that structure is useful for doing certain tasks. Okay, so tRNA is a very, very important molecule. Then in 1950, Alford and Martha makes genes from DNA. So they have done the experiments actually. Actually, they did this experiment. It's a very famous experiment. You know, Hershey Chase experiments. So they have made genes from, you know, makes genes from DNA. By the way, recently people are making proteins from non-functional DNA. What do you mean? What do you mean by non-functional DNA? So we have our DNA, as I mentioned, in human genome, we have 2% DNA which codes for proteins. Okay, rest is all architecture. So people are making proteins from the rest of the part as well. So artificial proteins you are making, and they try to see what we can do with that. It's a really interesting thing, you know, how this whole information is arranged. Then the year 1970. Why this year is very important? So, of course, I was born in this year actually, so I always remember, right? So anyway, so okay, before yeah, 1970 actually, yeah, 1952 to 1960. What so actually 1953, Watson and Crick. This is a very famous paper in Nature, right? Just around one page. If you can, you know, probably I can show you sometime. So they deduced that the double helical structure of DNA was discovered. This is something very, very, so, you know, there is a helical structure of DNA, and which you see normally in pictures, you know. So that was observed in 1953. So this was a very famous paper. And then George in 1956 showed that the site of enzymes manufacturing in the cytoplasm is made on RNA organelles called ribosomes. So ribosomes is again made of RNA. So again, you know, so basically, this is a important discovery of ribosomes in 1956. Important discovery of the structure of DNA in 1953. And of course, these are some sideline events. David Baltimore and Howard Trim, so they independently isolated the first restriction enzyme. What is restriction enzyme? By the way, what is enzyme? Enzyme is a protein. Okay? You might have heard, you know, take some enzyme, right? You are eating some enzymes, right? Some medicines in the form of medicines. So enzymes are nothing but proteins. What do you mean by restriction enzymes? Restriction enzymes are scissors. These scissors can cut the DNA at a specific position. Okay. So basically, if there is a DNA and you want to cut DNA at a specific position, it requires a specific scissor. So these restriction enzymes, if you can design, you can cut the DNA at a specific position. So these are very, very important discoveries, right? Restriction enzymes. Okay. So I hope you understand what is restriction enzyme. By the way, David Baltimore was, I mean, he got the Nobel Prize and he was president of Caltech for a long time. So I remember interacting with him, you know, in around year 2006 or 7, actually, you know. Anyway, so it's a nice person. Okay. So this era was really important, 86 to 1995, Larry Hood developed automated sequencing mechanism. You see, the journey of biology started with microscope, right? You have a microscope, you observe something, and then try to do experiments to isolate something. So initially isolation was for protein, right? Then slowly you started isolating RNA and so on and so forth, right? And then, uh, basically a lot of progress has happened in terms of experimental side. So we needed a sequencing mechanism. What do you mean by sequencing mechanism? Whenever we have this DNA or RNA, which is a sequence of, so DNA is a sequence of ACGT and RNA is a sequence of ACGU, right? And you want to read them, right? So initial reading was very, very difficult. Okay. So how to do this? Even nobody knows how to do it. So basically this automated mechanism of sequencing mechanism is an important discovery by, in 1986. And by that time, so many people were trying to discover portion of small organisms, say maybe a small bacteria or something, a portion of it, and they used to take a lot of time to just isolate and sequence and so on. It is at this point of time, uh, Eric Lander, I don't know whether you know, Eric Lander was a person who did PhD in coding theory, right? And he was looking for some position. So he started teaching mathematics at Harvard. Okay, and then one day, I think in one dinner or something, I remember a vague story anyway. You may find it on the somewhere, on the internet. So he started. So I think in a dinner or something, somebody asked him that uh, what you want to do. So he said that I have, what a specialist you are. So he said that I am a specialist in coding theory or information theory, right? So I want to understand how information is, when you transmit the information, digital information, or do something, So then the person told him that maybe you should look into the biology because biology has a lot of information. Okay, so he started reading about brain, basically neurobiology. Then he realized that there are, when I want to understand biology, I need to look at the genetics. Genetics is a branch basically where you study this, you know, hereditary nature of the species, right? Genes are there which are passed to from one generation to other generation, and then you can try to understand the relationship. So this is one side, and the other side is biochemistry. So basically, chemistry involves doing the experiments. These are two basically very, very important things in biology: experimental side with the biochemistry and the genetic sides. After the Mendel experiment, right? So these two were the side by side development. So then he started looking at it. Then he said, "Oh, well, if I have to understand biology, I have to look at these genome. And if I want to understand a genome, is basically the stretch of DNA, right? So every individual species has a specific genome. So how do we understand genome? So then he realized that biologists used to take a lot of time, to sequence. As I mentioned earlier, in 1986, already Larry Hood developed the automated sequencing mechanism. So whether robotics arm, etc., can be used. I mean, in those days, so can we expedite this process because it used to take a lot of time actually to do the sequencing at that in those eras. So he wrote I think a grant to the NIH, National Institute of Health, I think, which was initially rejected. He said that we want to sequence the human genome. Now you see, if you want to just sequence a small bacteria, it used to take a lot of time. So if you really want to look at the whole genome of human genome, which is around 3 billion base pair, kind of impossible task at that point of time. Also, people were a bit afraid to do this. Even biologists did not believe that it is possible. So see, this is the clear-cut example where you require interdisciplinary approaches, right? Because biologists are only, doing experiments. They are busy in doing the specific experiments and so on. And if you can actually use computer science or automated robotics, right? You know that robotics plays a very important role in your car assembly, for example, right? So you require a different kind of expertise to really do biology, right? So this is something at least then finally the US government was convinced, and in 1990, the 15-year Human Genome Project is launched by the US Congress. So this is a massive efforts of scientists from different disciplines and combined all over the world. And I think Eric Lander was so when the genome was sequenced, the paper first time published in 2003, he was the first author of that paper where, with so many authors, I don't remember, maybe more than 100 authors were there in that paper, something really, interesting effort, because it's the human genome, right? So nobody has done this before, reading the human genome and trying to understand things there, right? So then in 1995, there was a couple of other things, resolution maps of chromosome number 3. So as you know, there's that different chromosomes in human genome. So people started looking at a special kind of sequence, a specific sequence in the chromosome and trying to understand it. So I think people also realize that, for example, chromosome number 7, there is a P portion, Q portion. So in one of the portion, deletion of exactly 3 letters in the ACGT sequence gives you a disease known as cystic fibrosis. So basically, this is a lung-related disease. So genetic modification or errors in the human genome will lead to disease, right? So this is something important observation, right? So basically, okay. So this is something very, very important. Okay. So observation that certain diseases are possible because of errors in the human genome, right? So when the body is storing DNA and then you are trying to, so basically if some certain things are, so how this genome is altered, or you call it as a mutation. So there are different kind of mutations all the time. It happens inside the human body. Good mutation, bad mutation, right? So bad mutation will lead to disease, and good mutation will, make the species organism more strong, right? And so there's a lot of interaction with the environment as well. Anyway, so then in 1995 to 96, so 1995, Craig Venter first, sequenced bacterial genome. Okay, this was the 1995. So I think this was this was an interesting time, because I took admission in PhD program in 1993. So I started looking at development in biology at that point of time, because everything was so exciting. Another important discovery happened in 1995. Automated fluorescent sequencing instruments and robotics operations were introduced in the, as I mentioned, this was something which was missing in the sequencing mechanism, right? In fact, so normally the initial day of reading genome was, there is a stretch of DNA, and how do you read it? You chop it into different pieces, and then you see that if there is a specific sequence of ACGT, right, how do we read it? So they created an array kind of thing. So this mechanism is very popular, known as DNA microarray. So there is an array, it's kind of matrix, and you make certain dots. So of, say, suppose you want to read genome of certain size. So on each dot, you form an array, and so all possible strings of ACGT of that specific length you attach to each portion in the array. All possibles that are possible for h for n for specific things, right? And then what you do? So and these strings are known to you, that at the first spot we have A, suppose, right? At the second spot it is A, C, A, or whatever. So one bit you keep on changing and arrange all the strings on the array form, right? When you dip, so basically what you do, you take the genome, chop it into different sequences, right? Then amplify it. So you do a PCR mechanism. I will tell you about PCR soon, polymerase chain reaction, which is a way to amplify the genome. So you take a lot of this DNA, amplify it, okay, and then you dip this microarray into this solution. So what will happen? Because A is complement to T and C is complement to G. So whatever is known sequence on your array, right, at a specific spot, that will bind to the unknown DNA, right? So you will be able to read, because known string you know, and this will combine to unknown string. So you will read, so you know what is there at the unknown string, right? This is the mechanism through which this is a very popular sequencing technique known as shotgun sequencing. Okay, shotgun sequencing, you make pieces, amplify it, use this microarray technique to read the genome, right? And then obviously robots were involved in doing this business initially, right? So sequencing has made a lot more progress because of this human genome project itself. So the boom in the biotechnology industry came because of human genome, right? So many things happened, and that's why sequencing has progressed. That now you have pen drive kind of sequencing machine, which is known as nanopore sequencing, right? Sequencing means reading. Okay, this is something very, very important. Okay, so then in 1996, first eukaryotic genome is sequence was yeast. Yeast is another very interesting thing. Yeast you might be knowing, right? Why yeast is where yeast is used to to make breads and so on, right? So many things. So in Europe, especially, it was very popular, right? So in fact, yeast is the first organism for which we know the complete proteome. What do you mean by proteome? Complete set of proteins which are involved. So this is also very, very interesting problem. So you can look at any species and try to find out how many proteins are there. So initially, uh, people used to say that humans have hundreds and thousands of genes, means proteins. I mean, genes which can make proteins. So, but actually, but now we know that the number of genes are around 2,200 or something like that. So basically, the number of proteins that you can make, obviously you can make a lot more. So human proteome atlas, so human protein atlas, people started making. So the first thing is that you want to collect the data. And that's why the discovery of bioinformatics, right? So once you read this data, you require algorithms, fast algorithms, data processing techniques, searching, different, on different kind of fast machines. So that's why bioinformatics area started. Okay, so this is something very, very important. So I think I will show you. So E. coli is a bacteria, I think, which was sequenced in 1997. 1999 is the first human chromosome, number 22, was published. And I was, in fact, Nature published this sequence, this chromosome. I got a free copy from Nature because, I requested at that point of time. You can request because it's a special thing in the human history. Anybody can request the free copy. I mean, I think certain number of copies they distributed free. I still have that copy with me, from the Nature, So finally, 2003 Human Genome Project was completed, So this is a interesting history in the biology. After that, different kind of things. I'm not going to talk about other different aspects. Of course, DNA computing started in 1994, right? And that led to the DNA storage technology in 2012. You see, so everything is evolving now. When you're mixing computer science, maths, and biology, a lot of things are possible, and other aspects, right? Any question at this point? Now I will show you. This is a very basic picture. How many life forms? So one can ask this very basic question: how many life forms are there on this planet? How many humans, by the way? 800 crores approximately. How many ants? Some little ants. How many ants are there? 200 lakh crore. They are very, very important to actually create a balance on ecosystem of this planet. Otherwise, life is all hell. These ants are very, very, these are, very, very important. Okay. So let me show you some journey here. Again, a historical perspective. I mean, this is a very interesting book, What is Life by Schrödinger. So physical aspects of living cell. You see, so basically in those eras when the biology was starting, scientists and physicists, for example, Schrödinger was a very famous physicist. He wrote this. It's a very really interesting book. So now you see what is happening in biology, and you read this book. You can actually see a strong, thought process. Actually, what kind of things were there as a physicist, So how many life forms on Earth? Approximately 1 trillion. Okay. The order of 10 raised to 12. These many life forms are there. These are just approximations. How do you compute, right? How to calculate? Some way of, estimating. Not be accurate, right? But at least, in approximations, around 1 trillion life forms. This paper you can read. The scaling law predicts global microbial diversity, So how many plants? Well, there's, different plants, but here we are summarizing how many useful plants are there. 31,128 useful plants. Okay. So 139 vascular plants, complete genome. So now you see there's a lot of tasks for the humans. Look at all biodiversity. Look at each life form or each plants, flower, everything, and try to sequence the genome, right? Sequence them, create a database. So many people have published thousands of papers on this, right? Just read one, you have a machine now, read a genome, publish it, you get a paper, right? Read another genome, publish it, and you get a paper, right? So there's something really, really very interesting. So how many flowering plants are there? 369,000. So basically, the biodiversity is very, very important. If you take a flower or any plant, which is specific to a certain bio environment, so certain environment. If you change the environment, things will be different. So there are other parts of biology which are also making. So the environment plays a very, very important role. Okay, you cannot grow anything anywhere. Okay, there are different conditions for growing. Okay, so now I think this is also pretty old, something around 2016 actually. But out of these useful plants, 17,810 used as medicines, human food 5,538, okay. Fuels 1,621, animal food 3,649, all kind of things, even social uses. Indian medicinal plants are around 5,000, I mean, this is not done by me anyway, right? These are all collected from different reports, right? So, but what I'm saying that this is something really interesting to know, right? And one should know that how many of these things are available, right? How many bacteria, by the way, on this planet? So if you assume weight of one blue whale is around 140,000 kilogram. So all the bacteria on the planet are equivalent to 50 million blue whales. So much bacteria. And this bacteria is playing a very, very important role. We are alive because of this bacteria. Because these bacteria controls the weather system of this planet, Right? So these small organisms do a lot of useful things for us. Okay. Now you see that there are so many bacterial infections. By the way, why do we have bacterial infection or viral infection? Anyone in the audience can tell me why do we have bacterial infections or viral infections? What is the role of these microbes, viruses or bacteria? Anyone? Why do we have these infections? You know infection happens, right? Infection hua, go to the doctor, take medicine, and that's all, right? And even you should know how to, yeah. Huh?
Body is not ready to, as in the changes which are made due to these bacteria. So maybe other bacteria are not able to counter it for the normal temperature, and so that is a reaction.
Maybe they enter our body through air track or anything, and then they start reproducing and feed through.
I think both of the answers are not entirely correct. Let me tell you. So each of these microbes, viruses or bacteria, they have friendly ecosystem where they grow, and they are not harming the species wherever they are, right? So you will see that there are many bacteria inside our body which are useful, right? Similarly, there are many bacteria on maybe elephants or any other species, right? So what I'm saying that different species carries their own microbial ecosystem. Okay. Now what happens whenever these things changes the species? then there is a problem. Something which is friendly to, say, monkeys, if you switch to human, it will be a dangerous thing. And the same thing happened with HIV. HIV early form was SIV virus, which was friendly to monkeys, right? So there was an encounter with humans. It changes the species from monkeys to humans, and then it became deadly for humans, right? So whenever you change, because then what happens? This species wanted to survive, so it will make changes inside their genome, just struggling, trying to survive in the new ecosystem, and the new ecosystem is also fighting, right? Our immune system actually fights all the time, and there's an attack of different viruses, microbes all the time on us, but we are fighting all the time, right? So this happens all the time. Now what happened whenever we have these things, whosoever wins will win the thing, right? In the COVID-19 case you have seen, right? So origin of these microbes, viruses is very, very important. They will come from some source, you know. I mean, these are some list of certain, bacterial infections. So, in 1654, again, so using handcraft microscope, bacteria was discovered. So they call it as animal clues, tiny animals, You can see that. It's an interesting thing, right? Now, I'm going to tell you one very interesting story. Because of bacteria, the entire trade was possible because of bacteria, right? All the economic development, and actually, as I mentioned many times, that the last 40, 50 years was because of economy grew out of internet. But now the next 30, 40, 50 years is only for biology. If you don't know biology, it will be difficult for you to survive in the economy, right? So in the next thing, this is very, very important. Let me show you this story of ships. There is a bacteria, cyanobacteria in the ocean, So basically, it controls the temperature. Basically, you can think of this that, because of it, that ocean has cold water, hot water, right? Those kind of things. And, all that interaction, I mean, all that is controlled by these bacteria, okay? And so because of these bacteria, so, warm air will go up and so on. And so the whole ecosystem of, basically currents basically are formed on the planet. So you can see one picture of these currents here, ocean current. These are mentioned, And discovery of these ocean currents was very, very important when humans started, going here to one place to other place using these boats, right? They discovered that if boat is in one of these currents, it's very easy to travel, right, from one place to other place, and that resulted in the whole technology development in that area, creating bigger boats, because now you don't require much fuel. Basically, just using with the currents, you can actually go to a longer distance, right? And that has resulted in people started moving, the industrial revolution in Europe is because of this only, because many people from Europe started traveling different parts of the world using, ships. Before that, it was through land, and the land is, is not very uniform everywhere, right? So there are difficulties. But so that one of the Silk Route is from India to, western part of the India. It goes to the longer, to mid part of the earth, right? So basically, these are very, very interesting thing, right? So these ocean currents started I would call it as the ocean economy, right? The whole new things started. Surat has one very important, port, I think, right? So at that point of time, early, a lot of trade used to happen from Surat actually. So it is. Sorry, I don't know why there are. Okay, so we call it as the age of. We can call it as the age of discovery. 15th century to 17th century, all these big boats, with all this kind of technology, they used to travel from one place to other place. Maybe I think I will stop here, because I think I will take some more time to complete it, right? So next time I will complete this biology story tomorrow, right? what are the possibilities, okay? And by the way, some of the material is there. You can actually just read it, okay? Or watch the videos, okay? Thank you.